starting with this semester, we stopped sort of last semester. We left off with evolution and how sort of scientists have looked at how things have been passed, or how offspring develop traits from their parents. And the natural progression of inquiry goes from, well, what's actually passed on? Chromosomes. Figured out as chromosomes. Okay? And now the natural progression of inquiry, now we end up with the fact that chromosomes are composed of protein and DNA. So what is passed on? DNA and protein. Which one of those is responsible for the traits? And that's sort of where we are. Okay? So, we know, you know, that DNA is the genetic material. All right? So keep that in mind. That's where we're going. That's your result. All right? Where I'm going to take you through sort of the progression of how scientists saw chromosomes are passed from one generation to the next, to figuring out that it's really DNA that's the important part. Okay? So those steps in between chromosomes and figuring out DNA is where we're going to talk about today. Okay? So we know that, you know that, who came up with the structure of DNA? Watson and Crick, right? We know from last semester, you should remember DNA is a double helix, it's a nucleic acid, it's double-stranded, right? That's the basics of DNA structure, all right? You're going to get into how the structure of DNA is related to its function in a little bit, of passing material down, how it's replicated, how genes are passed on. All right, we'll get into that. All right, you know that DNA is the substance of inheritance. It's passed on, it's the genetic material. But what you don't know, may not know yet, is exactly how scientists came up with that result. Okay? And you know that genes are encoded in DNA, and that's the heritable information. So I'm giving you sort of where we left off, our starting point, and the result, DNA being the genetic material, how we plug in the steps in between. Okay? So here's a picture of Watson and Crick after they figured out the double helix and somewhat stole it from Rosalind Franklin, or stole her data without, I shouldn't say steal. They didn't really steal it. They just sort of used it, not necessarily with her permission, and came up with the actual double strand. Won a Nobel Prize and are forever immortalized in science. Okay? So, early in the 20th century, we figured out the molecules that are actually passed on are chromosomes from one generation to another. Okay? And chromosomes are made up of DNA and protein. So figuring out what the traits are that are passed on from one generation to the next was sort of a question. Well, is it protein? Is it DNA? They weren't really sure. Okay? So that's sort of was the challenge of the questions to biologists in the 20th century. All right? So Morgan, his group showed that genes are located on chromosomes, okay, by doing the tagging that he did. And the two components of chromosomes, DNA and protein, became candidates for genetic material. So now scientists are challenged with devising a way to determine which one of these, DNA or protein, is definitively responsible for passing on genes. Okay? And there are two really good experiments classic experiments that are used to define this, right, at this time. And so the key factor in identifying this was using selecting correct organisms, right, and selecting correct genetic mechanisms that allow this to occur. All right? And this first was discovered in bacteria and viruses. Why would a geneticist or someone who's looking at looking at inheritance, utilize bacteria and viruses as a model organism. What's the benefit of using bacteria?
Easy to grow.
Is that?
Easy to grow.
Easy to grow. Okay. What else? They're pretty simple organisms in terms of their genetic background, okay, or their DNA structure. We probably know the best, most about them right now in terms of organisms, right? Think about the early 20th century, we haven't really utilized yeast or eukaryotic cells yet. What else is happening in the early 20th century that is pretty important? Historical lesson. Early 1900s, lots of people died.
But yellow fever.
Not yellow fever. Hmm? Happened. Millions of people died in Europe because of this before World War I. The bubonic plague. Okay? The plague, all right, Black Death as it was, is passed on from bacteria. Okay? And several French scientists are working on figuring out what the causative agent of the plague is, and what to do to help people. So, and you may have heard of Jonas Salk. You know who that is? So every one of you have gotten a vaccine, right? Yes? You can thank him for that. Okay? Louis Pasteur, right? Pasteurization. These are scientists at the time that are actually working on bacteriology. Okay? So bacteria is sort of the model organism of the time, not eukaryotic cells, not cancer cells like they are today. Okay? So bacteria and viruses were mostly studied because of their relation to medical importance. Okay? So what did it do? All right? So this was an experiment by Frederick Griffith in 1928. All right? Griffith worked with two strains of bacteria. Okay? One pathogenic and the other one harmless. All right? So we'll minimize this now. Put that off to the side. So here we have, we'll call them S-type bacteria. All right? When I take these bacteria and I inject them into a lovely little mouse. It's not the best looking mouse. Okay? All right? When I inject them into a mouse, because they are virulent, they cause disease. What happens to the mouse? It gets sick. And eventually, it dies. Okay? That's the S-type, or the virulent strain. Okay? The other strain of bacteria, we'll call them R-type. Right? These are non-virulent bacteria. A related strain to the S-type bacteria, but they don't cause disease. Okay? So now I do the same thing. I take the bacteria, I inject them, and I'll try to draw a better mouse this time. Okay? Little whiskers. All right? I inject them into the mouse. What happens? Nothing. It lives. It overcomes the bacteria that's been injected. Big deal. Okay? Now, the next thing he does was he takes the S-type bacteria, but it has been heat killed. All right? What would happen to the bacteria if you expose it to high heat? What's going to denature? Proteins. Okay. Proteins are going to denature. So now I take this bacteria, right, and I inject it into a mouse. What's a little fatty mouse? Okay. It's heat killed. The proteins are heat killed. Okay. What happens?
Mouse gets sick again.
Does the mouse get sick? No, the mouse does not get sick. Okay, so he's okay. All right. Now, the next thing is I'm going to take and mix the S-type bacteria. Okay? And I'm going to mix the two, the S-type strain and the R-type strain. All right? When he mixes the two, all right, so when he mixed the living cells, the harmless, some living cells became pathogenic, transformation. This is what happened. Okay? So S cells, this is a little better rendition of it. Okay? So now when he mixed the heat-killed S-type cells, which should have killed the proteins, right, with living R-type cells, which are non-virulent, the mouse dies. And he goes back and he looks. He takes what they would call a necropsy. All right? They go back and they basically cause, it's an autopsy in humans. Look at the dead or the organs of the dead mouse and take a cultured bacteria back out, and he shows that he finds living S cells. So in some way, those R-type cells that were living here, right? The R-type cells that were living here picked up a virulent something or other and transformed. That's transformation. Okay, this is what he called transformation. In some way, these R-type bacteria, these non-pathogenic bacteria, picked up something from the heat-killed S-type strain, and then they changed. They changed their phenotype in some way. What conclusion can you make from this experiment about what the genetic material is? Think about it. Talk to your neighbor about it, and come up with an answer. What conclusion could you make? If the glove does not fit, you must acquit. What's that from? O.J. Simpson. You guys were like, what, 2? 95. It was 95. It was the summer of 1995, and then I remember when the verdict was read in October of 95. I was a college freshman sitting in the Union watching it. All right, so what conclusions can you come up with? Abby.
Since the proteins were denatured, the genetic material wasn't passed down.
All right, so it wasn't passed down from protein. Can you make the conclusion that it was DNA? Yes or no? No, because you haven't definitively proven that it was DNA, but you have disproven that it is protein. Okay? So it's not protein that's been picked up from here. Because what else could be picked up? RNA, could be DNA, it could be carbohydrates, could be something else. All right? But this is the process of transformation. Okay? We know now that it is DNA that is picked up. And in lab next week, you're going to do transformation of bacteria. You're going to take exogenous DNA, mix it with bacteria, and bacteria will pick up that DNA and integrate it into their chromosomes, and it will be expressed. Okay? So, this is Griffith's experiment about transformation. Questions on that? All right? Later work basically showed that the transforming substance was DNA, and we'll get into that. However, many biologists were like, no, there's a running debate. I don't believe you. The stuff is it could be RNA. Something was wrong with your experiment. Right? They fought a big bicker about stuff, us biologists. Mainly because the structure of DNA hadn't been figured out yet. Okay. Second, we haven't come up with a mechanism of how DNA replicates. and how genes are structured in DNA. So there's lots of questions. Mainly, at this point, it was mainly proteins were the best guess. Okay? So, experiment in working with bacteriophage. All right? What is bacteriophage? Anyone know? Bacteriophages are viruses. Okay? They are viruses that specifically target bacteria. Okay? So it's a phage virus, what biologists term phage, right? But it infects bacteria. Okay? And a virus, in its most simple form, is basically a nucleic acid, DNA or RNA, okay, and a protein coat that protects that genetic information, right? And that's in its simplest form, right? When we talk about viruses, we'll talk about more complex viruses than bacteriophage. So the two things that biologists are arguing are the genetic material actually make up viruses, or actually make up this bacteriophage, protein and DNA, all right? And a virus, you'll learn in a couple weeks or more in depth that virus must infect cells to take over the cell's metabolic machinery in order to produce more virus. So what does Hershey and Chase, or how does a virus work, or bacteriophage? All right. So here's a bacteria. All right, and along comes a lovely bacteriophage. All right, and so what you see in red there is the protein coat of a bacteriophage, and this is the lovely, in blue, is the genetic material, the DNA. Okay, the way that a bacteriophage works, it attaches to the outside of the cell, of the bacteria cell, and injects the DNA into the cell. Okay? What the protein coat is not injected into the cell. Okay? So, nucleic acid gets in, protein stays out. What do you think, is this set up for a good organism to use to tell which one is protein or DNA is the genetic material? Yes or no?
Yeah,
Yeah, why?
because it's only letting the DNA into the bacteria.
All right, so only the DNA gets into the bacteria, so if the bacteria become transformed in some way, what does that tell you?
That DNA is the genetic.
DNA is the genetic material. Okay? And that's basically what Hershey and Chase did. Okay? So if there was a way that you could follow where the protein and the DNA went, you could tell whether or not the DNA got into the cell, or if the bacteria became transformed, right? Or if the protein got in the cell or didn't get in the cell. All right? And this is what Hershey and Chase basically done. Elegant way. There's your bacteriophage, scanning electron micrograph, right? Your phage head. Inside is the DNA that's protected by the protein coat. There's just tail sheath, tail fiber, and this is actually DNA being injected into the bacteria. Okay? Just all right. So Hershey and Chase, all right? They've definitively shown that DNA is the genetic material of a DNA phage known as T2. So there are different types of bacteriophages, some of which utilize DNA, some of which use RNA as the genetic material. Luckily for them, they found one that would use DNA. All right? So they designed an experiment showing that only the DNA of the T2 phage and not the protein enters the E. coli during infection. All right? So I gave you the sort of how a bacteriophage works. Right? They didn't really know the outline of how a bacteriophage infects a bacterial cell. So this was part of their experiment. The experiment was set out to prove two things: how a bacteriophage infects E. coli, and second, what the genetic material is of that bacteriophage. Okay? And so their conclusion was that the injected DNA of the phage provides genetic information. So now I'll show you the sort of how they did this, and that was their conclusion. See if the experiment, okay, you can come up with the same conclusion from the experiment. Okay? So, oops. All right, so here we have this batch 1. Okay? So, when you add a little bit of bacteriophage to a culture of bacteria, what's going to happen? I add a little bit of virus to this culture of bacteria, that the virus will infect the bacteria and produce what? A lot more viruses. Okay, so they used that concept, and in the media of that bacterial culture, they also added a radioactive isotope of sulfur. Okay, so they used S35. So now, as the phage are infecting bacteria, they're producing more bacteriophage. What are they producing? What's the coat of that bacteriophage? Protein. Okay? In DNA, is sulfur found in DNA? No. Is sulfur found in proteins? Yes. All right? So this is a way of labeling the proteins only. All right? And not the DNA. Okay? So they let that go for a while. Lots of phage are produced. All right? Then they break all the phage down and separate it out. Okay? And what do they look at? What do they look at? They say radioactivity, phage protein is found in the liquid. Okay? Centrifuge to form a pellet, and they look at, right, to find, here's the bacteria that are down the bottom, and the phage are in the suspension. What does that tell you? What did this centrifuging do? Or centrifugation, proper word. What did that do? What's the purpose of spitting this out?
Separate the bacteria from the viral proteins.
So you're going to separate the bacteria, the stuff inside the cells, from the stuff that's outside the cells, from the proteins. Okay? And so now they found all the sulfur, or the labeled proteins, are outside of the cells, right? And none of the labeled proteins are inside the cells. Okay? So protein doesn't get into the cell, right? Does that definitively say that DNA gets in? No. So they have to do a second. Okay? Second experiment is here. Okay? They do the same thing at the beginning. DNA, or you have your bacteria, add the phage. Instead of labeled or isotope of sulfur, they use an isotope of phosphorus. Why phosphorus? Phosphorus is going to be found in DNA. And phosphorus is not very abundant in proteins. Okay? So it's a way of selectively labeling DNA. So you have the radioactive DNA, you add the phage, okay? Break it up, do the same centrifugation, and where is all the radioactive phosphorus? They're in the pellet, meaning it's where? Inside the cell. All right? So the DNA gets in, the proteins stay out. These two experiments together, right, led to and then follow it up with the structure of DNA. Watson and Crick led to the idea of DNA being the genetic material. All right, and then for decades that was sort of, that's it. And then later on we find that RNA can also form genetic material as well. All right, questions on the Hershey-Chase experiment and conclusions that can come up from that. The Griffith experiment. All right. Any other questions? You understand how you can make those conclusions? All right? All right, good. All right, so we know that DNA is made up of nucleotides, right? So here we have DNA, right, composed of nucleotides. Ooh, I'm out of practice, so the handwriting is going to be pretty bad right now. Okay? What are nucleotides composed of? Three things. A nitrogenous base, so a base. Okay. What else? A 5-carbon sugar and a phosphate. Right? What are the bases that are found in DNA? Cytosine, guanine. Okay? Adenine, thymine, cytosine, and guanine. Now, Chargaff was looking at the composition of each of these nucleotides across species. Right? So see if there's a difference in DNA across. Well, all right, fine. What he found out and realized that there was always an equal percentage of adenine to thymine. And there was usually, or for the most part, always an equal percentage of cytosine to guanine. This came to be known as Chargaff's rules. That in whatever percentage, say, in humans or in Drew's DNA, for instance, there is 30% cytosine, which means it has to be how much guanine? 30%. And then based off of that, how much percentage would you have adenine and thymine? 20-20, right? That's the rest. Right? So it's 40% divided by 2, right? So because they're paired together. So he came up with this across species and found that across species, a percentage of DNA of a particular base across species may have been different, but within that species it was always pretty much the same. Or cytosine always equaled guanine or was very, very close to it. All right? Same thing with adenine and thymine. This led to the step of base pairing, right? Led to the conclusion that adenine pairs with thymine and cytosine pairs with guanine. This was another conclusion that Watson and Crick utilized in order to come up with their double-stranded anti-parallel structure of DNA. All right, so Chargaff's rules, all right, percentage or composition of DNA. All right, questions on Chargaff, Hershey-Chase, Griffith, etc. All right, we'll stop there for today. On Monday, we'll go over what the structure of DNA and sort of lay its groundwork for its function as a genetic material. Lecture will also be posted, the recording of the lecture will be posted on Moodle this afternoon.